Hello, explorers, and welcome to Reach the World's Explorer program. For 25 years, Reach the World has inspired youth to become curious, confident, and compassionate global citizens through virtual exchange. My name is Tim, and I'm so glad you're joining us for today's live stream event with the Cleanest Air on Earth expedition. And we have two very special guests for today's event. We're traveling to Fort Collins, Colorado to meet Dr. Jessie Creamean and Chelsea Bekemeier, both at Colorado State University. You have come to know and adore explorer Kelsey as she has braved the Southern Ocean to collect samples and data. And today, Dr. Creamean and Chelsea are going to show us where those samples and data go after the collection phase and what they do with these valuable clues. As always, don't miss Kelsey's amazing virtual exchange articles from the Research Vessel Investigator, which you can find at the link in the chat. I'm delighted to welcome a number of classrooms who are joining us live today or watching the recording after we're through. Some of those amazing classrooms are the Palmer Public School, Toluca Lake Elementary, Ensign Elementary, Julian Carter School, Franklinton Elementary, Bolts Middle School, Blevins Middle School, Lesher Middle School, Cache La Padre Middle School, Lincoln Middle School, and several others. We're very, very glad you're here today or watching the recording after the fact. For those of you who are tuning in via YouTube, please feel free to use the YouTube chat bar to let us know you're here, where you're joining from, and of course, to share any questions you have for Dr. Creamean or Chelsea as we go. We'll get to as many questions as we can in the next 45 minutes. And without further ado, Dr. Creamean and Chelsea, welcome to Reach the World.
Hello. Hello.
Hi, welcome. So glad to have you here today. We're getting a little bit used to seeing Kelsey aboard the ship and sort of popping up in surprise locations at sea. So it's noticeably different. We're someplace very different today. Jessie or Chelsea, whoever wants to go first, can you tell us where you are today?
Sure. So I'm Chelsea. I'm one of the grad students working under Dr. Creamean, and this is one of our main labs. So this is where we bring the samples back from the Southern Ocean and Antarctica, and we often freeze them, and then when they're ready for testing, we'll bring them out and prepare them in our hoods, and then we run them through our ice spectrometer, which are the nice little cardboard box-looking things that we have right here.
All right, fantastic. I can't wait to get a closer look at that. It's a really exciting opportunity for students to be inside a professional laboratory and see what working scientists are doing in those spaces. So this is very exciting. Jessie, would you like to introduce yourself and tell us a little bit about yourself as well?
Yeah, I'm Dr. Jessie Creamean, and I'm a research scientist here at Colorado State, and I am the principal investigator for the project that Chelsea is working on and Kelsey is working on with Kelsey's supervisor. And I actually have mostly focused my work in the Arctic, so I was involved with a lot of expeditions there, but now we're transitioning to the other side of the world and doing similar measurements down there.
Fantastic. Can we pause for just a moment and define what is a principal investigator? It sounds very important. What is that?
So basically, we're the leads of the project. So we seek out grant funding from places like the National Science Foundation, and we do research projects with them to try to understand some scientific gaps we have in our understanding of the Earth system.
All right, very good. So last time we talked to Kelsey, she was floating in the Southern Ocean. She is now safely back in Perth and sort of unloading her materials and some really valuable data and samples that she's collected as part of her cruise. And all of that is being packaged up to be sent primarily to you, right, in Fort Collins. So maybe we can just dive right into it. I'll put myself backstage and you can show us what's going to happen to some of those air filters. And there were some water samples and a bunch of things that she's collecting as part of this expedition once it reaches you in Colorado.
Well, first, before they even get here, I'm actually going down to Tasmania to get the samples in a couple weeks and send them back so Chelsea here can start analyzing them with our system. So why don't you go ahead and explain what we're doing with this?
Yeah, absolutely. So all of our samples come back in these actual super cool cryoport. So they're really good to transport all the way from the Southern Ocean back to Fort Collins, Colorado. We take those samples out and we often store them in a freezer to make sure that we maintain preserve the samples before they're ready to be taken out and experiments performed on them. Typically, we'll take out the samples and we'll do things like pull the filters and suspend them in water. And then once we suspend them in water, we shake them to get the ice nucleating particles out. And then we'll either run that through our ice spectrometer to see how many ice nucleating particles are within that sample, or we'll perform different tests on it, such as heat to get rid of any biological material in the samples, or hydrogen peroxide to get rid of like anything organic, to see what parts of the ice nucleating particles are going to be dust or biological material or other organic materials in the samples. And the ice spectrometer, is essentially a fancy word for we pipette all of our samples into a tray, and then we have really super cool air and nitrogen running through, so it's nice and dry and cold. And we're gonna take snapshots at each point in time of when it's coming down in temperature, and we're gonna see when those samples freeze. And then we use math to kind of back out what fraction of that sample are ice nucleating particles that are activating at different temperatures.
And I guess
should we go ahead? Sorry, no, go ahead.
Yeah. So ice nucleating particles is a term that y'all may not be familiar with. So these are really important in the atmosphere because they are basically these little specks of material high up in the air, and they're really important for forming clouds. So if you think of a cloud kind of like a tree, it needs seeds to grow on. And so these special little specks of material actually help ice form in clouds. And so the ice is important because it can change how the clouds interact with sunlight. It can change how it interacts with heat that's trapped in the Earth's atmosphere. And it can also affect how those clouds rain or snow out after. So they're really, really important specks of material that are in the air that often you can't even see with your naked eye.
Amazing. Could we ask you to sort of go on the move through the lab and show us a couple of things you talked about? I know that for some of the students who are watching, maybe who have gotten into some early lab sciences, know what a pipette is, for example. But I think it would really help to see some of those things.
Over there, maybe? We can start.
Yeah, we can start.
Okay, sorry, I'm doing this from this weird angle, so sorry if I miss the frame.
So this is an example of one of our hoods. It is a laminar flow hood, which is a fancy word meaning that it consistently puts out air that's fairly clean, so we can make sure that when we're working in the hood, all of our samples are clean and there isn't dust collecting on it, other materials that could settle on the sample and affect the measurements that we would see. So in here we have our different pipettes that we would use based on what we need to put in the sample. And then we have our DI water, which is water that is essentially deionized or all the impurities taken out so that we can make sure that there's nothing in the water that would affect our ice nucleating particle fraction as well. We have our rotometer, which is one of the ways that we kind of resuspend the particles once we take that filter out and we put it in water. And then we have that going for about 20 minutes to help resuspend those particles. We use aluminum foil, which looks really silly, but it actually is a really good, clean material that doesn't attract ice nucleating particles. So that's why we use a lot of aluminum foil in the lab. So not just good for baking, also good for science. Sweet.
So we'll come inside. We'll try to show you the inside of one of the ice spectrometers.
So this is our ice spectrometer. It does have a penguin on it because we're representing our affiliation with the Southern Ocean in Antarctica. If you open this, you'll look inside and you'll see that we have our trays. We have our trays inside, and we have a plate on, or we have this glass plate on top. We have a camera attached at the top that actually takes pictures every 18 seconds of those samples, so we can see at what times and temperatures that they freeze. And these fancy insulated pipes are going to be carrying that nitrogen gas through there, so it maintains a really dry and cold environment. And then, of course, we have our laptop. We can show them this one if you want, that's open. Do you want
Oh,
to?
sure. Yeah. Hopefully you all don't get motion sickness. I apologize.
So yeah, you get motion sickness like this other notion. So this is what our interface is going to look like. So once we put the samples in, we'll have it running on here, and you'll be able to see, this is a plot of temperature versus time. So you'll be able to see the temperature decreasing with time. And then these little dots represent all of the different wells of the samples, and they'll pop green once the sample freezes. So ideally, when we're towards the coldest point at the end of our time frame, so near like 30 degrees, minus 30 degrees Celsius, many of these will hopefully be green. Yeah. Anything else on that?
So nice work.
Yeah, amazing. It's so cool to see how you are looking at things, particles, elements that are basically invisible to the bare human eye. Are, you know, if you didn't know better, you might assume they're not there, but you're identifying and better understanding things that are completely invisible to people. What is the hardest part about what you do with the samples? Is there something that's particularly challenging, or is there something that is really easy to get wrong, or might, if you don't do it carefully, it produces inaccurate data?
Yes. So from my perspective, so typically we'll take the filters and we'll suspend them, and then we'll run a sample in the eye spectrometer without doing any treatment. So we just get like a big spectrum of, oh, there's X amount of these ice nucleating particles. And then kind of what I was talking about earlier, if we want to back out what portion of this is dust or what portion of this is biological material, we have to run additional tests. One being heat, where we just heat it up basically to where it's boiling for 20 minutes. And then the other is going to be a hydrogen peroxide treatment, which is hydrogen peroxide and heat. And we suspend it in hydrogen peroxide, which really likes to denature the proteins and organic material inside. So kind of like kills the organic or biological stuff. For me, that is the hardest one, because it could take anywhere from 20 minutes to like an hour, depending on how much stuff you have in the sample. And also you have to be very precise, because you're using a tiny amount of, I don't want to get too deep in the weeds, but once you stop the reaction with something called a catalase, you have to be very careful with the amount of catalase that you're using to stop that reaction. And so that can be difficult. And in fact, I had to work pretty hard with our postdoc students, which a postdoc is someone after they do their PhD, they'll work in another lab for like a year or two before they move on to get a faculty position. So I had to work with them to like really get that timing pretty correct. And I would still say that I'm still learning.
I really like the laboratory phrase, denature something.
Yeah.
It sounds like sometimes you need to denature in order to measure something else that's not nature-based.
Yes. Yes.
Dr. Creamean, can you talk a little bit about like a career in a lab, in the laboratory sciences? This is your job. You work at Colorado State University and you work in a laboratory space like this on expeditions and collecting data and solving big complex questions and problems. How does somebody start in a career like this, and what is a typical day like for you?
Cool. So my path was a little unconventional. I actually wanted to be a medical doctor first. And so in college, I was on the pre-med track, but then I took a physiology class and I got my first C. So I was like, oh, maybe I shouldn't be a doctor. But I was taking chemistry classes at the same time because that was my major. And I got really interested in it. And then I took an environmental chemistry class. And that's when I, believe it or not, back then started to learn about climate change and why it's so important. And so I was like, oh, wow, I want to work in this field. Like, I want to do something about this. So I went to grad school and I worked for an atmospheric chemist. And so we did a lot of research in California in the mountains there, actually looking at these specks of material called aerosols in the air and how they affect clouds and climate. And so that kind of got me down the climate science realm. But then to get into the Arctic realm and working in a lab like this, I did a postdoc fellowship like Chelsea was talking about at the National Oceanic and Atmospheric Administration in Boulder. And that's where I really started to learn about how climate change is affecting the Arctic and the Antarctic much faster than the rest of the world. And so, you know, thinking of these poor polar bears and penguins that live in these regions, I really wanted to focus my efforts there and my career in that trajectory. So 10 years ago, that's when I finished my postdoc, and I've been doing Arctic and now Antarctic science since, and working in this kind of lab setting has been really fun. So Chelsea said you can go on to teach at college when you're done with your postdoc, or you can actually become a researcher in a national lab like I did initially, or you can be a researcher in an academic lab like this one. And I really like this environment because it gives you a lot of flexibility to research really key, challenging questions. And so a lot of my projects, I have about 8 in total right now, are focused on, you know, what are these little specks of material that help ice form in clouds, these ice nucleating particles? Where do they come from? How do they affect clouds in the Arctic and the Antarctic? And so these things can come from the ocean, they can come from permafrost, which is soil that's frozen in the high latitudes, that's thawing because of climate change. They can come from trees, they can come from plants. Chelsea mentioned that they're biological, so they can be things like fungi or pollen or plant bacteria that get into the air. And so these things are really important, and we have a pretty poor grasp on them, especially in the polar regions. So it's really fun working in this setting. I'm not much doing the lab work anymore. That's what Chelsea does mostly, but I help with making sure the project, you know, works smoothly and do all the reporting and make sure Chelsea gets awesome papers publishing her findings out of these kinds of things. So it's really fun to kind of oversee this whole project from beginning to end and do that continuously.
Excellent. Well, thank you for that. Palmer School, I know we've got some earth science students watching today. I'm going to come to you next for some questions. If anybody has any questions to ask, and Mr. Lee's class, if you have any questions, be sure to add them to the chat. We'd love to fold them in. I want to ask the same question though to Chelsea, who's at a different stage, like maybe slightly different interest or a different path to where you are in your career right now. How did you get there? What interested you about this and made you want to pursue this path?
Yeah, so I was kind of like a born nerd, so to speak. I was really interested in science from a young age, and when I was young, I was like, oh, I'm going to be an astronaut. I knew I wanted to be in science, but for me, it was actually kind of also not a linear path. So when I was in like middle school and stuff, I actually struggled in math a little bit, and I wasn't the top of my class in math anymore. And so I kind of lost hope that I would be good at science. So I got a degree in a different field, and then I decided to go back to school later for science once I was more confident in myself. And I think that that's really powerful because a lot of people kind of in science second guess their ability. But as long as you try it and go for it, you can definitely like make it happen. And so, yeah, I went back, got another degree, and now I'm in grad school here.
All right, excellent. That raises the question. I know you just came from class. What class did you have today?
So I had 2 classes. I had one called Objective Analysis, which is essentially data science for people in atmospheric science. And then the one I came just out of was Responsible Research, which teaches you how to be a responsible researcher. And it's for anybody that's like working off of government grants, that we make sure that we're truthful and have a commitment to open data so that everybody can view it, and make sure that it's a really fair, equitable process for everyone involved.
All right, wonderful. Well, I'm going to bring Palmer Public School onto our camera here. Hey, Palmer, how's it going? I know these are earth science students, so lots of interest in the earth and the big questions related to the earth. I'm going to turn it over to Palmer. I know you've got a couple questions. If the students who are asking questions wouldn't mind stepping up to the camera so we can see and hear you a little better, that would be great. Otherwise, I'll turn it over to you. Fire away.
All right. Mason, do you want to get us started?
Sure.
All right. That'd be awesome. Stand up and introduce yourself. My name is.
Hi, I'm Mason Lockwood. My question is the lab equipment that you have on the ship more or less advanced than the equipment that you have in Colorado?
I'll take that one. I've been on a ship a lot. It's less advanced. So we sometimes take a scientific instrument that's similar to what this does, but it's more portable. This one, so we showed you kind of the main components of it, but in order to cool it down, it requires a really big, heavy chiller that basically recirculates cold fluid through it. So that's really hard to transport into the field and work on a ship, and especially ships in places like the Southern Ocean where you can get almost 100-foot swells. It can be a little challenging to work in those environments. So a lot of the efforts on these ships are focused on making sure we get the samples. And sometimes we do some initial tests on the ship to make sure our sampling is sufficient. But for the most part, we save the detailed stuff for back here.
All right, great question. Let's roll right into a second question from Palmer, if you have one ready to go.
Hi, my name is Jack, and my question is, have you recovered any data or samples from the Southern Ocean yet?
Yes, so part of CASO, which is the broad project that has different voyages that Reach the World is featuring, we had an initial voyage called SOTS, which was the Southern Ocean Time Series, which was in May of this year, or sorry, it's already 2024, in May of 2023. So almost a year ago, and we already do have most of those samples back, and we're still waiting on some, but we have recovered and already done the treatments and processed the samples from that initial voyage.
Yeah, wonderful question. We haven't gotten too much into it, but the Cleanest Air on Earth expedition is just one cruise as part of a long-term study, as Chelsea mentioned. And Chelsea, I think you're heading out on a ship in the future too, right?
Yes, so I'll be on the Denman voyage starting February 2025.
All right. Are all of those cruises answering similar questions or attempting to answer similar questions?
Yeah, I will say that the reason we have so many voyages is a part of this big project is to kind of double the amount of observations that we have in the Southern Ocean, because since that area is historically not the easiest to get to, and also this is kind of a newer field of research, we don't have good observations currently. So there's a big unknown as to like what the ice fraction in clouds are over the Southern Ocean and what impact that has for our radiative balance and climate models. So that's why there's so many voyages going on as a part of this project, is we're really trying to double the amount of the observations.
Okay.
And they do kind of go in slightly different locations. So most of them leave from Tasmania, which is even below Australia. But some of them are going to Antarctica. So like the one Chelsea's on is actually going all the way down to along the coast of a glacier in Antarctica. Some of them are kind of sticking around Tasmania and Australia. So they're trying to cover a wide spatial area too.
All right, excellent. Great question. Great answer. Palmer, while we have you on, do you have one more question you want to ask? The floor is yours. Oh,
Don't be shy. We don't bite.
Excellent, excellent.
All right, my name is Brody. What ways of transport will the samples take to get from the ship to your lab?
Yeah, sure. So as Chelsea mentioned, we use these special shippers that stay very, very cold with liquid nitrogen. And typically it can take up to 10 days, sometimes longer. But the travel part's actually not the longest. So planes fly pretty frequently between Australia and the US, but the longest part is actually getting the samples across international borders. We won't bore you with the details of international shipping and customs, but that it can be challenging to get samples from different countries to the country here. So we have to make sure it follows all kinds of permits and appropriate paperwork, which blah, 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 boring stuff. But we usually make it work and they can get here within a week or two.
Jessie, you mentioned that you are traveling to Australia. Why do you have to go? Is there something that you're doing special to package them up, or do you bring some back with you on your person, or what role do you play in the transporting of those samples?
Yeah, so I'm going to make sure that they are packed nice and snug in those special shippers and then send them off to the US. And also, because the next voyage that Chelsea is going to be on isn't for another year, we're actually going to pack up all of our equipment that's currently on the Investigator, ship it back to the US, do kind of some maintenance, make sure we have enough sampling supplies for the next voyage. So basically packing stuff up and making sure it gets safely back here.
Right, excellent. A long trip to pack up some
Yeah.
samples. How long of a trip is that? Have you made that journey before?
No, this is the first time going to Australia and Tasmania, and I have to fly from Denver to LA, and then LA to Melbourne, Australia, which is a 16-hour flight, and then Melbourne, Australia to Tasmania, which is another few hours. But I'm hoping I see, some wallabies or Tasmanian devils to make up for the long trip down there.
Excellent. And that in a nutshell is why Kelsey's not with us today. We started doing the time zone math to see if she could join us, and it will my mind just couldn't already where she is. So
I think they're like 16 hours ahead of us here in Colorado. Yeah.
Amazing. Well, Palmer Public School, great questions. I don't want to take you off screen before everyone who wants to has their chance to ask their question. Are there any other takers?
Probably not, Tim, right now.
All right, very good. Well, thanks a lot for joining us for those great questions. I have a couple of questions for you, Jessie and Chelsea, that I want to ask on behalf of other students. Do you have any favorite facts about the atmosphere? I've personally learned a lot from you about how the atmosphere is influenced by not just what's up in the air above us, but the ice nucleating particles that come from the water. The atmosphere is impacted from lots of different directions, not just the air. Do you have any favorite facts that will surprise us about the atmosphere? Maybe something that most people don't think too much about.
Ooh, that's a tough question. There's so many cool things about the atmosphere. I just heard a statistic recently. It was how much does the atmosphere weigh? Do you know that? It's an absurd amount of weight.
Yeah, if our bodies didn't have the ability to regulate equilibrium with the atmosphere, we would be crushed by the atmosphere.
There's so many gases and particles and specks of material and water in the air that it weighs some crazy amount that you wouldn't even think of. It was insane. Sorry, I don't remember the actual number.
No, that's a good fact, because if you're not talking to atmospheric scientists like yourselves, most people I think would say it weighs nothing. It's air. It's like light. It's all around us. But interesting to know how heavy it is. Is there anything in particular that one or both of you are looking for with this fresh set of data coming from Kelsey that she's been collecting over the last few months? Is there something you're super excited to dig into and look to see if it exists in this group, or if something that you've seen prior or anticipate seeing is present?
Yeah, sure. And Jessie can correct me if my hypotheses are wrong.
Hey, the hypotheses.
But so it's been summer in the southern hemisphere, and so it'll be interesting to get some of these samples back. I did see from the chat, because there's a chat that we have with the crew that's on board, and they were very excited to see high levels of dimethyl sulfide, which is kind of a result of biological material like phytoplankton releasing those gases into the atmosphere, and that can help form different types of secondary aerosols, and seeing other types of organic material because it is summer in the Southern Ocean. So I I don't know, I'm personally excited to see the levels of IMPs and run my test to see like what fraction of that would be biological, because since it's summer, we might be expected to see more biological sources.
Yeah, I would say I'm most interested in seeing how things compare to the Arctic. So a lot of my work has focused in the sea ice. So I've been on icebreakers really far north, close to the North Pole. And there's actually a lot of small critters there that are active during the summer. So phytoplankton, there's algae in the ice, and those feed the little tiny shrimp that are in the water, and those tiny shrimp feed the fish. So they're really important for the food chain, but they're also important for climate, as we've been talking about in clouds. So I am curious to see how things are different or maybe similar to the ice regions in the Arctic. So do we see these same organisms down in the sea ice in the Antarctic waters versus up in the Arctic? And are those as ice nucleating or not compared to what's in the Arctic?
Do you find having processed data like this, and especially Dr. Creamean yourself, like having worked on a couple expeditions, that the more data you get, the less questions you have, or the more questions you have?
I would say more. You can often, like Chelsea was mentioning, you have a hypothesis or you have a scientific question that you're like, we need to find this out. It's really important to help improve our climate models, that help improve how things might change in the future. And so sometimes you can answer those questions, or you can at least like improve our knowledge a little bit on them to help improve the models. But you see things there that you're like, whoa, I had no idea this was going to happen. So we've seen phytoplankton blooms that have created aerosols that generate INPs, these ice nucleating particles that affect clouds in the Arctic. And it's just, you see these things that you're like, whoa, I didn't know this was going to happen. It's really, really fascinating. So there's new discoveries that open up new questions for sure.
All right, fantastic. I want to make sure I'm getting all my questions answered here. Good question coming from students. We have seen Kelsey collecting the data. We've seen the lab on the ship. We've been to the Southern Ocean with her. We've now been to your lab. We see where those samples are going. You are learning a lot about the samples that are being collected and will be collected in the future. What do you do with that knowledge afterwards? What's like is there a final step after the laboratory analysis that you do to sort of like tie a bow on what you found?
Yeah, so we do the lab research, and then we process it, and then we take what we get from the post-processing, and we either test our hypothesis. Like, we can either say, yes, we see significant like rise in biological sources during Southern Ocean summer, or I could say, no, there's really no difference between this and the one SOTS, which was more like their winter. So we just use that, and we kind of test our hypothesis, and why did we either reject it or reformulate it, and then we essentially write a paper about it. And that's what goes out into the world, and that's how most science is done. So hopefully this will become a paper.
Or more.
Yeah,
Yeah, these papers are in scientific journals, so they're kind of meant more for a science audience, but they can be really interesting, and you can read some pretty kind of bigger picture stuff of why we're doing this. And we're also going to be working with a climate modeler who's going to take our data, and she's going to try to improve how the models make clouds behave. So try to get it closer to the observations by using our data.
Wow, very, very cool. For all the students who are watching who may be familiar with writing like a lab report after they've done a project in school, how closely does what you write or what you produce in a written form after this complex and much more involved series of like hypotheses and data collection and data analysis, how closely does that resemble a lab report, maybe just in form or format that students might be familiar with?
I would say that at the end of the day, it is pretty similar. It's just like a very high level way of applying the scientific method and writing lab reports that you do in high school. It's just definitely at a higher level. So you You have a hypothesis, you have objectives, you have research outline, you have your results, you have whether or not you could reject or accept your hypothesis, and it's very similar at the end of the day.
All right, fantastic. Well, I have learned a lot today, and I know our students have too. I'm going to go and say ice nucleating particles to everybody I know and impress them with my new knowledge of these super particles that are on the cutting edge of atmospheric research. So thank you both so much for sharing your time with us today. We've got some Palmer students still backstage. I'll bring them back on camera just to say goodbye and thank you to Dr. Creamean and Chelsea. Thank you all so much for being here. Thank you to our online audience. Can't escape without saying that it is International Women's Day today. And on behalf of Reach the World and all of our students, you two inspire us. So thank you so much for the cool work you do and for taking the time to share it with us and explain it. I know you're doing it and thinking about it at a very high level, and it takes a lot of skill to translate what you're doing into a way for non-scientists to understand. So I appreciate everything you do, and hopefully we can connect with you again sometime down the road. Maybe as Chelsea goes out into the field and is collecting new data, and we continue the Cleanest Air on Earth expedition. All right, so yeah, with that, that is of course all the time we have today. Thank you again to our YouTube audience for joining us. Please don't miss Kelsey's amazing and wonderful articles on the Cleanest Air on Earth expedition journey page. Again, I put that link in the chat on the YouTube. We will hopefully see Kelsey again soon now that she's back on dry land. I think she wanted to do another call when she's back in Colorado and maybe a little less far away from us, and we can reflect on the experience of going out into the field and being at sea for so long and doing her amazing field work. But until then, so long for today. Keep exploring the world around you. I hope we'll see you all back here for another Reach the World live stream event. Thanks so much.
Thank you.
Bye-bye.